Hi everyone, welcome to this edition of our Livino Virtual Data Science Seminar Series. We're delighted today to be joined by Leo Martins, a tenure track fellow in the Department of Engineering, School of Engineering.
School of Engineering, Department of Electrical Engineering.
That's what it is. Where he's a tenure track fellow, and he's talking today about his work in describing the evolution of genomes through Bayesian models. So when you're ready, feel free. Please.
All right, thank you. Yeah, I have no idea how long it's going to take, so yeah, just stop me if I'm running over. So yeah, I think I chose a very aggrandizing title, but actually the talk is going to be much simpler than that. I'm going to talk a little bit about phylogenomics and phylogenetics and a few Bayesian models that I developed in order to solve this problem. And by the way, I did a few years of postdoc, and then I worked for many years as a staff scientist. So I was a senior bioinformatician, and so I was developing models while applying them to microbiome, to bacteria work. But what we're going to see here is all over the place. And yeah, so first I'm going to give, I think, a 5-minute introduction to phylogenetics and phylogenomics. So what is phylogenetics? Well, phylogenetics is the problem of describing the evolution of living things. And so how do we do this in practice? Well, in practice, we have genomics. And so genomics data, here I'm representing each genome by one number like C29, C43, C8. So these are different genomes. And there are some techniques that we use to find where the genes are in those genomes. So, for instance, in this case here, we can see that C29 we have TSSM, and then TSSL, TSSK, and there's an order for them, which is indicated by there. We don't need to worry about that. The important thing for us is that we can find in different genomes we can find the same or very similar genes. So, gene is it's that specific region in the genome. And then once we find those genes, and then the next step is to find what they call the site-wise alignment. So homology is the concept that homology means that two things came from one in the past. And so previously, when we were looking at the genes, we were finding the genes. So we were finding the homologous genes, which means things that when you look at the genome, we assume that they came from in the past, they came from the same thing. And now that we know that these things, we assume that these things are from the same thing in the past, that they are homologous. And then now we use an optimization algorithm to try to optimize every site in the genome. So every DNA site column, we try to optimize this the alignment of them. So this is also a homology problem, and we're not going to look into this. But as you can see, there's already some problems there, right? So depending on if I call something a gene or not, or if I say that they are aligned in this way or in a different way, so they're assumptions of homology. But let's forget that for a second. Let's assume that we can do that. And so in phylogenetics, what people work 99% of the time is what they call multiple sequence alignments, MSA. So this is the alignment of all the sequences for one genome. And then we can see here, so Classel, Muscle, T-Coffee, and MAFT are different programs, are different algorithms that are used to optimize the alignment at every site. And in this paper, by the way, they were saying that, well, depending on the algorithm that you use, you have different answers, and these different answers might have an implication. And so, but usually biologists forget about this. They use very quick and very well-known algorithms, and we assume it's known. And then now comes phylogenetics by itself. So phylogenetics is, as I said in the beginning, is describing the evolution of the living things. It's describing the tree. So it's inferring which tree they came from. And so there's some classes of algorithms. So in this case here, I'm representing a hierarchical clustering that if we have two genes or two genomes, and then we can calculate the distance between them. And actually, if we can calculate the distance between any two things, we can start clustering them. And so the last step of the clustering is going to be this dendrogram. And so we can assume that the datagram represents the evolutionary history. Sometimes we can do this, sometimes we cannot. And for these cases, we also have parsimony and likelihood methods. I'm not going to describe all of them to you, but it's just to say that some methods, we know what's the optimal answer, and some other methods, they are heuristics. So we have one tree, and then we ask, is this tree a good explanation for the data that I have or not? And then you try a little bit different tree, and then you ask again until you find a tree that you're satisfied. So parsimony, likelihood, and Bayesian models, they are based on that. And there's the fourth step. So this is where I think we spend a lot of time playing with, which is phylogenomics, or let's say post-processing or phylogenetics. So once you have the tree, once you have a good description, a reasonable description of the species, and then you can start asking questions. But when did this happen in time? How long ago? And in this case here, I'm showing an example that is very dear to my heart, which is phylogenomics. So phylogenomics means if you look at different genes, because as I said in the beginning, we find different genes, and then for each one of these genes, we can do this thing. But if you look at different genes, these different genes, they might tell you different stories. And in this paper here, so this paper is from 2017, and they're already saying, because this is an ongoing fight in the evolutionary biology community. So if the porifera are the sister clade to Mammalia or Gnetophora or something else. And so in this paper, they say, well, it depends on the genes that you choose. So some genes, and so each one of these of the columns here is one different study. So different studies in the past, they use different data sets. And so depending on the data set that you use, the data set will say, well, 50% of the data is going to give you this T1. And sorry for those online. So the red one, the red bar says, well, 50% or something close to 50% supports T1. But actually there's some 20-25%, which is the green bars there, that support T2. And then there's some other genes that support a different tree. So that's the problem of phylogenomics. Phylogenomics is when you recognize that there's variability within the genome. So if you look at different, if you ask what's the story each gene is telling you, they are going to tell you different stories. And so this is very briefly, so the two main ways of that people came up with to solve this issue is supermatrix and supertree methods. I worked a lot with supertree methods. I don't think supermatrix. I work a lot with supertree methods, and supermatrix are not my favorite, but both of them are quite well used. So supermatrix basically says, well, if you have a bunch of genes, then concatenate all of them, and concatenate all your data, assume that you have one big block of data, and then you estimate one tree, or you use Bayesian methods to estimate your distribution of trees. Supertree says, no, actually, if the different genes are telling you different stories, then you ask, what's the story each gene is telling you? And then later, once you have these inferred trees, and then you try to compare the trees. And yeah, this slide, I think we don't need it here, but it's just because this is a very broad. So it's just describing what's Bayesian statistics. And in the case of phylogenetics, so we want to know the probability of a tree given our data, and then this is proportional to the probability of the data given the trees and the prior over the trees. And I'm going to show you very briefly, instantaneously. Yeah, and Bayesian models, as we all know, we have explicit models for uncertainty, so we are interested in the uncertainty, and we can use very, very complex models because the computation doesn't change that much. So yeah. I'm going to show you very briefly. This is going to be the only slide where you actually see what the likelihood of a tree looks like, so the probability. So what it's saying here very quickly is that the probability of observing T in 1, A in 2, and A in 3, the ones that are marked there in blue or green, is going to be given by the probability of a T becoming, of an A becoming an A on the T2, which is between 2 and 4, probability of an A becoming an A at T4, so it's over that branch length, times the probability of that A at the internal node 4 becoming a T in T1 plus T4. And so you have to sum up overall internal nodes. So this becomes cubically complicated as we calculate that, because we have to think if you're not working with DNA, if you're working with proteins, then instead of 4 you have 20, or 64 if you have codons, or if you have arbitrary models, and then you can still use likelihood, but you know you have a state space there, and you have to compare all the possible internal assignments for that. Why do we need phylogenomics? Why do we need trees? Why do we need evolution of these things? I realize this is a bit off topic, but so I'm gonna just skim through it. So one case, this is what I used to work a lot. It's unpublished. I don't think this is ever gonna be published because the main author is doing something else now. But this was a very interesting case that we had samples, we had pathogens sample from the live pigs, and then when the pig goes to the market, and then we have a pork, which is written there as pork. So this is the pork meat. And then we also had the chicken, and we had a chicken meal. So if you buy in the domestic supermarket, and this is also interesting because although I'm Brazilian, I was not involved in the collection, but this is data from Brazil and the UK. And then we asked it, so can we find genes that are associated to pork or to pig or to chicken? And you know, this is a linear regression, and linear regression, when you take into account the specific form of the phylogeny, it has a name. So usually people call it it's the PCM, the phylogenetic comparative methods in general, and in the case of microbial bioinformatics, people call it bacterial GWAS because it looks like the human GWAS. Anyway, to make a long story short, it's possible. So once you have the tree and you have the genes, if the gene is there or if the gene is absent from that particular sample, and then you can ask which genes are associated to this or to that. Another case is a critical view of microbiome classification. So as I said, you go to the market and then you take the pork meat, and then you check which pathogens, which bacteria are there. You're going to have a lot of bacteria. So how do you know exactly which bacteria are there? There's this very famous gene called 16S, and this is because it's very cheap to sequence. So you don't need, you don't need a PhD. You don't need a whole group to sequence and to get this. So it's a very easy, let's say, straightforward technique based on this gene and this gene, and then you look. Oh, in my sample, I have, I don't know, 10 copies of the gene from E. coli and 10 copies from Salmonella and 20 copies from something else. So you can say, well, this we have 10 and 10 of E. coli and Salmonella. And then in this paper, we said, yeah, but this is a very short gene. And so if you use different genes or if you increase the length of the gene, you might have different answers. And for classification, which means to know, to describe, say, it's E. coli, it's Salmonella, or something else. And then you need larger genes. And so basically this is what we said. And we said, well, if you don't have much money, so if you need to use the same arsenal as we use for 16S, perhaps if you use 16S and 23S, which is a neighboring gene as we see there at the top, you have a better signal, phylogenetic signal and the same classification. And that's because of paralogs. So as I said in the beginning, well, to make a long story short, you have homologs, which are everything that you assume came from a single ancestor. But sometimes you have duplicates. Sometimes you have several copies in your genome. So you have several things that look like they have the same ancestor. So which one of them is? Well, probably all of them are, because the genomes are complex. And so in this case here, it's because 16S is one of those genes that you can find more than one copy in the genome. And so depending on the copy that you choose, you're going to have one answer. So this paper is not mine, and it was published during the COVID years because they said, well, if you since we have so much data for COVID, and then we have very beautiful trees, very complete trees, and with these very complete trees, we can actually find when there was an importation event, when the sequence came from a country, and we can pinpoint. And basically this is because if you look at the tree and then you look back into the tree, the trees, you can have timed trees in this case. So we know this, the time where the sequences were sequenced, that were sampled. So for instance, in the case of Japan, I think everyone arriving in Japan, they had to have had a COVID test, and then if it was positive, they would be sequenced. So, and the UK as well, I think the UK was one of the ones that sequenced most. And anyway, so since you have a lot of data and you know exactly when they arrived and when they were sequenced, and then you can trace back in time, even if you don't find that ancestor sequence, and then you can say, well, somewhere, I don't know, 2 weeks ago, in the flight between Brazil and the UK, someone in the airplane got infected. And then we use this rationale, and we develop fast algorithms to describe when this happens. So we call this the migration patterns. And in this case, I think we are describing the beta and the delta variants. Now everything is Omicron. Now Omicron is everything we hear about, but in the beginning we had this other variants. And this is another case. It's the same story. So once you have the tree, once you have a lot of sequences with the data, with the dates, and then we can actually try to estimate when the things happen. And if we see at the bottom, so here I'm zooming in the exports and imports, so we can see, let's say, a more uniform influx of imports. So there was a lot of people bringing SARS-CoV-2 into Lebanon, and then at some point in February, you had a lot of people exporting. That's because a few weeks later, we found the same one or a very similar one outside of Lebanon. And I'm just gonna skip this. This is just say, if you don't use the tree, you can classify things. You can find antimicrobial resistance genes. That's what AMR stands for. Sorry, usually I put all my acronyms. But yeah, so we can find antimicrobial resistance genes, but they are not one-to-one associated to the strain or to the species or to the genus. So, you know, if you use the 16S that I mentioned a few slides ago, to say, well, you have E. coli, and then just telling that you have E. coli, and even if I tell you the strain of E. coli that you have, sometimes it's not enough to say if you have some antimicrobial resistant genes or not. And that's because here we are not using the tree. So my idea for the future is to use trees to describe these things. Okay, so I have 28 minutes. So I'm going to describe to you two challenges. So now that we saw what's phylogenetics, what's phylogenomics, we see phylogenomics again in a bit. And why we need the trees, I'm going to give you two challenges that current techniques, or, you know, before we tackle them, there were no good solutions. And so the first one is viral recombination. So this is an old problem. Now with SARS-CoV-2, people start looking into it again, and now it's much easier in the sense that we have much more data. And so if you have a lot of data, none of this is a problem. We don't need Bayesian models. If you observe everything, you know, and every step of the way, every day you sample everybody. But if you don't, if there are gaps in your knowledge, and then that's when you have to take the uncertainty into account. So I'm going to give you one example. So this is when I left Brazil, and then I had some colleagues in Brazil, and they saying, "Hey, Leo, there's we think all the HIV that is in Brazil came from one or a few viruses, and it's called CRF14, I think." Because they all look like this, they look like a mixture of subtype F and subtype B. And subtype F and subtype B are very old sequences. I think they were sequenced in the 90-something. And they were shown not to be recombinants. But then when you look into Brazil, all the sequences, parts of them look like F and parts of them look like B. I'm going to describe this very quickly because I think we might be familiar with this. It's a sliding window approach. So if we have the F sequences that we say these are the blue ones, and then we have the B, subtype B sequence that we say these are the red ones, and I have three query sequences, that's what the question mark stands for. So I have three query sequences. You know, three people went to the hospital, and then I have these three sequences there, and then I ask, okay, let's color them. Let's see what's the mosaic, what's the recombination pattern of these three sequences, and then you just go as in a sliding window through the genomes. And then you say, well, in the beginning, they look like F, and at some point in the middle they look like B, and then they look like F again. And so we do this for all of them, and they all look the same. Does it mean that nothing happened there? Does it mean that it was one BF that was infected somewhere, as we know nowadays happens sometimes with SARS-CoV-2? The same individual, it's infected by 2 different strains, and then this individual probably is immunocompromised, so he keeps the SARS-CoV-2 inside them for a few weeks, and then recombination happens, and then when they distribute this new variant, it's a recombinant. We don't know. So we know that they have the same color, and in HIV, I don't know nowadays if they still call, but they would call this CRF14. Well, they all look the same. They all have the same mosaic pattern, which means that it's color pattern. But probably there's more going on there. So for instance, what I'm showing here, I don't know if you folks can see, let's say the leftmost tree and the rightmost tree, they are different, which means that although they all look blue, the trees are different. So there's something happened there that is not described only by the blue one. And so I spent some time working on how to calculate the distance, how to describe. So if you give me any 2 of these trees, and then I was developing algorithms to describe exactly what happened in terms of recombination, which is equivalent to horizontal transfer, if you're more familiar with that in microbial world. So yeah, we said, well, they all have the same mosaic structure, but do they share the same history? No, there's something going on there that we cannot know. And the solution that we thought was to use the phylogenies. Instead of just coloring them blue or red, we actually use the phylogenies, and then we model the similarity between neighboring sites. So we say we calculate the distance between the trees, and this distance must be biologically motivated. As I said, the number of recombinations or horizontally transferred. And so yeah, this is how I came up. This was the alternative title for the presentation. So Bayesian regularization of phylogenomics, or phylogenomic regularization. So these are regularized methods, regularized in the sense that, if you apply a regression and then if you increase the number of data and you increase the number of parameters, then you have a collinearity problem, and then the number of parameters might explode. And then you have to regularize. You have to say, no, actually, I know that most of the things don't change. Most of the things are the same here, are 0 in the coefficients in my regression. And so we have to use a distance that represents something biologically, and then we use this distance explicitly in the model. So this is what the model actually looks like, and not exactly, but a representation of it. So we say every site, and by the way, when I say site, is a column there in this, it looks like a feature matrix, right? And in the end, so every site, every feature in this feature matrix has a tree, and this tree potentially is different from the next one, from the neighboring ones. So it's a different parameter. But we say, but actually, although these parameters, they can be different, the probability of them being different is lower than the probability of them being similar or the same. So we say, we assume that and then in the end, we have these different colors there. So we assume that most of them are going to be the same, unless you really need a different tree to explain that difference. And so in the full model is going to look like something like this. So the probability of my parameters, which are the 3 and other parameters that I'm not interested now, given my data, and my data is this multi-sequence alignment that looks like a feature matrix, it's going to be proportional to the likelihood. So I think the term at the top is the likelihood. So it's the probability of each one of the columns of the data. So I is the columns, given the tree and given other parameters, times some priors. And one of the priors, importantly, let's see. Yeah, one of the priors, importantly, is what we call it the penalty. And I think now, this is a regularization term. In the papers, we call the penalty, because we penalize distances that are non-zero. So we say the probability should be the same. Sorry, the probability should be high if the trees are the same, and the probability should decrease if the distance starts decreasing. So which means that, for instance, if the green tree and the orange tree, they are very different. In terms of recombination, you need a lot of recombination to describe the difference, then the probability should be low. And so you only observe green being different from orange if you actually, the data tells you strongly that. And as I said, I spent quite a few time working with these distances. So the distance that we found for this case is called the SPR distance, the sub-tree prunary graft distance. By the way, this is not very easy to calculate, but we found a good approximation, and we show you here. So this is the number of SPRs, the sub-tree prunary grafts. It's a graph modification of the tree that we apply, and then here at the y-axis, it's the estimate one. And so the blue one, which is the one that we develop, is good at least for when the distance is not very large. But if the distance is very large, then you need a lot of recombinations. Then you say, well, look, the probability is going to be low anyway. And the RF means Robinson's fold distance or symmetric distance. So this is the symmetric distance in graph theory. So it's the number of edges that are unique to one of the trees. And this is by far, 99% of the times, by far the most commonly used tree distance, the Robinson's fold distance, because it's very easy to calculate, it's very fast, and you can implement it very easily. But for our case, it's not very good, right? Because it reaches the asymptote very quickly. And yeah, so here I'm describing exactly what a sub-tree prunary graft is. So assume that you have the leftmost and the rightmost trees, and then you want to describe how many SPRs you need. So you prune 3 and 4. You prune a branch from 3 and 4, which is the red one, and then you plug it again, you regraft it on the branch leading to 1. And so if you do this movement, this SPR, the sub-tree prunary graft movement once, you can describe the distance, the difference between them. And so if you need to do it 4 times, and then that's going to be your SPR distance. Yeah, on real data, it worked well. So this, we simulated a very short alignment, so only 512 base pairs. And we knew, because you can simulate data given a tree. And then we simulated these 128 base pairs for each tree, and we know exactly what happened between the trees. So for instance, the first tree and the second tree can be described by an SPR of 2, can be described by 2 recombinations. So if you remove 3 and plug somewhere else, and then if you remove S8 and plug it somewhere else, you're going to have the second 3. And then from the second 3 to the third one is the same. If you remove S7 and plug it somewhere else, and then if you remove S9 and plug it somewhere else, and so forth. And then our method not only detected the location of these breakpoints, but it also detected the number of recombinations. And then we applied to our regional data from South America. They all look very similar. The state-of-the-art method, which is also Bayesian, but you can use it only for 4 sequences. It tells the color, the mosaic pattern, but we found many more recombinations that don't change the color. So there are potentially recombinations between the blue ones or between the red ones. And we could use all the data at once. And as I said, the state-of-the-art software at the time, it could work only with up to 4 sequences, which is the simplest tree that you can have on rooted tree for that is different. Okay, so that was the first challenge, and that's how we solved it. And I said, well, perhaps we can use it for other stuff as well. But before that, just in a few minutes, I'm going to describe what's the general idea behind phylogenomics. I already mentioned this briefly, but the idea is that gene family, so if you look at different regions in the genome, they're not going to tell you the same story. And so that's what we say here, that the gene family tree is not the species tree. So the species tree is the grand tree that we all think of when we think about, let's say, the tree of life, or if we assume there's a tree, right? That's a different question. But anyway, let's assume there's a big tree, there's a single tree of life. Each one of the genes should not follow that tree, because there's biological processes there that make them different. So for instance, one of them is paralogy that I mentioned briefly. So when you have different copies of the same thing, in the bottom here, at the bottom, I'm representing a genome where we have chromosomal duplication. So we have a lot of genes being copied. And so the genome there, it has 2 copies of TSSH, which is the red one, 2 copies of TSSL or I, TSSC, and so forth. So, several of them, you have 2 copies of them. And this is a case of paralogy. And you can have missing data. I don't know, perhaps, the person doing the sequencing couldn't find with the specific that the machine was broken, or they didn't use the proper equipment. Anyway, so but there are biological reasons, as I said, for that. And the biological reasons are gene duplications, losses. There are population processes when you remember that you actually don't have one individual evolving. You have population of individuals, and this population of individuals, some of them might go to the mountain, some of them might go to the river, and then they are free to speciate. So, and then they reproduce, and then you have more and more of these individuals. And lateral transfer of genes, horizontal gene transfer. This is a big issue in microbes. So as I said, the AMR, the antimicrobial resistance genes, virulence genes, they don't follow the tree of the species because they follow other trees, and that's because there's a lot of horizontal gene transfer in bacteria. Anyway, so there are a lot of biological reasons why we shouldn't expect the same tree to be observed along the genome. And as I said, I think I said this again, I should have removed it. And so we go to the next challenge. So the next challenging is, so how do we describe then the evolution of the gene families? By the way, I say gene family because usually when we say genes, people think about the orthologs, you don't have paralogs, you have only one copy of that thing. Or if you have more than one copy, you forget about it and you assume you only have one, like the case of the 16S. And when you say gene family, you make it explicit that you're talking about everything that might have common ancestry, even if they were paralogs in our immune system. Actually, mammalians, they have a lot of duplication. So we have different copies, and these different copies, they are free to evolve a bit differently. And they're important for us. So, for instance, if I have this bunch of trees here, can we guess what's the species tree? What's the answer is no. I don't want you to start thinking about it. So these are fungus, and I went to a database, so the database that I was working at the time, and then for this particular database, all the paralogs are there. And so we can see that some gene families, they are, so let's say the blue one, the light blue, they have only a few species represented. I'm going to move here. I don't know if people online can see. So this one here, we don't have all the species represented. We have only a few, but I think we have one copy only of each. So this would be a tree that everybody would be happy. But for instance, the leftmost one here, we have, so we have some paralogy here. We have the same name more than once. I think it's Rhizopus is one case. No, Aspergillus. No. Yeah. Anyway, Schizosaccharomyces is Pombi. Yeah. So we have the same one here at the top and at the bottom. So why is that? That's because you have one genome, and in this one genome you have 2 copies of the same. It might be, it might be a bit different, but with the algorithms we can say, well, actually they are the same gene, they have the same function. And so we have, 2,000 of these trees. And can we find what's the species tree? What the species tree is that represents all this mess? Well, we can use regularization again. Remember, we can use distances. So we say I like species trees, the S one there, that is close to all my gene trees. And what do I mean by close? Well, close in some biological term that I cannot define, but we can discuss. We can think about several distances. And then if I say, well, the probability of the gene family tree having been generated by that species tree is going to be proportional to how similar they are. So for instance, if the distance is 0, then it's very probable. If the distance is very high, and distance, it just means that they are biologically very different. and then the probability is going to be low. By the way, why am I all fuzzy talking about the distances between the trees and shaking my hands? And because there's no Euclidean distance between trees, right? There's no single way of describing. And so depending on your problem, so the number of duplication losses can be very different from the number of horizontal heat transfers between, if you're going to explain using one or the other. And yeah, so again, we have a likelihood method. That this likelihood method, it's going to be it's not exactly, so this is not exactly the likelihood, but let's skip that for a minute. So we have that the posterior probability is going to be the likelihood times a penalty again. And this penalty is the probability of the gene and the species tree, but it's not exactly what I wrote there at the top, because as I said, I don't know exactly what are the distances that I want to, I don't know exactly what's the distance between them. And so this is 80% of the answer. So the answer is that there are fast ways of calculating the distance between a gene tree and a species tree. And most of them are called reconciliation distances. So for instance, if I use a population, I'm sorry, I'm not going to describe all of them, but it's just to say that if I use a population approach, then it's one deep coalescence, or and if I say, no, actually what's happening here are duplications and losses, and then it's going to be, I can use basically the same algorithm, just change a little bit, and it's a very fast algorithm. And then you can say, well, actually one duplication and 3 losses is all you need to describe the difference between the blue one and the red one. And then we can use some stochastic distances, like, as I say there, the Moore-Robinson's folds distance. Anyway, there are different ways of calculating the distance, but we can calculate using all of them. And here are some other distances. So the Robinson's folds distance, as I said, is the number of unique edges, and so we see the ones in red if I want to calculate the distance between them. The mass distance is the maximum agreement sub 3. We don't need to worry about that, but it's just to say there are a lot of distances that we can, even if I don't know biologically what they mean. What does it mean to have a Robinson's folds distance of 6? Is it biologically much different from having a Robinson's folds distance of 1? I don't know, but we can add them all into the algorithm. And now we are ready to write what's this probability. So yeah, this probability is going to be a multivariate discrete exponential distribution. There's a partition function here. You see it took 1 year to calculate that, but anyway, it's just to say it's feasible. So we can add how many distances we want, and then someone comes to me and say, no, actually, you know what, we are working with bacteria here, and in bacteria we don't have duplication and losses, so remove those distances. And then yeah, we can remove them, and then we can run the software again and see if we can estimate what would be the set of species trees that are consistent with what we observe in terms of the gene families. I'm just already giving the answer here. Okay, so then I went to a database. By the way, this database I was looking for it the other day. It's defunct now, so it doesn't exist anymore. 3FEM. So 3FEM used to have the trees for proteins, and it's part of EBI. So I think you can still find, but now it's a bit hidden. And then we asked, okay, give me the biggest trees that you have for Drosophila, for fruit flies. And then, as you see, it gave me 43 trees that look like this one at the left. You see the same color again and again. That means that there's a lot of paralogs there. There's a lot of, so you look at the one genome, and this one genome, they have a lot of copies of the same gene. I don't remember exactly which gene is that, but anyway, there are 43 of those. And then we used only those to estimate. So now, what would be the species tree of Drosophila given these genes? And then it gave us this tree, and apparently it makes sense. So even the routing makes sense. I mean, I'm not a Drosophila person, so I cannot tell you exactly what if it's perfect or not. But I think it was in agreement with what people knew, and the routing as well. And the routing was an interesting one. And a few years later, then people start publishing papers saying, yeah, actually, if you use this kind of methods, you can find the routing. Because if you change the routing, by the way, the root means that special node there at the leftmost side of the tree. So you can read from the past at the left to the present at the right in that case. And so they said, well, if you change the routing, if you have the wrong routing, if you have a species tree with the wrong routing, the apparent distances are going to be larger than if you have the correct routing. So the correct routing, in a way, minimizes the distances for duplication losses distance, for this reconciliation distances. And this was a big thing because it's not very, you need extra information to find the routing. Usually the trees, I think I had my students there. I forgot to shout out. Yeah. So my students here, they are telling me that all the phylogenetic trees in general, they are unrooted. So although it's much, much easier to read and to represent them as rooted, less having this special root node, which is like a dot at the leftmost of each tree, in practice, they are not rooted because of the way the likelihoods are calculated. Anyway, so usually we don't know what's the routing of the trees, and we need some extra information to find that. And this is one of the ways. And this is another thing that I was also very happy about. I'm sorry, there should be a transition. So forget that you're seeing the answer here at the bottom part. So the inputs. So this is a two-stage MCMC. So you use an MCMC. You estimate the Bayesian, the posterior distribution of each one of the genes, and so you have a distribution of trees for each one of your gene families. And so you give this bunch of files to the software, and then this software is going to try to reduce the uncertainty. And we were interested in the species tree, in the distribution of the red tree, but it also gave us very good recalibrated, let's say, distribution for the gene trees. So we gave a gene tree with a lot of uncertainty, and then we asked, and then we throw a lot of these to the software, and then the software said, "Oh, actually, you know, this you had a lot of uncertainty, but actually, I can reduce the uncertainty because you have information from the other gene families and from the species tree itself." So the result is what we see at the bottom there. So at the left, which should be the cyan, the light blue, is the input frequency of the true tree, which it was around, it's less than 0.2%. So we give a bunch of trees, and let's say 10% of the trees are the true one, because everything is simulated, so we know what's the true here. And then the output is going to improve the frequency on each of the gene family trees. So we are very happy with that, because even if you're not interested in the species tree itself, some people say there's no tree of life. It's a network of life. You know, there's no tree, and they are right in a sense. So what's the correct tree? Perhaps there's no correct tree because it's a composition of trees. It's a graph that cannot be represented by a tree. So yeah, even if you're not interested in the species tree, you might be interested in the gene families and the evolution of each one of the genes, and this kind of method can help you improve. I only have 2 minutes left, and this is a very, I don't know if it's helpful or not, but anyway, so this was a specific case where this is fungal trees as well. I think we have 2,000 trees. And they are very well behaved trees in the sense that we can calculate the Robinson's fault distance, the H distance, the SPR distance between all pairs. And so, you know, if you have a matrix of pairwise distances, and then you can do an MDS, and then you can look at how they look like in 2 dimensions for each one of the distances. That's why we have 3 plots there. And then I asked the software, can you find me trees that are good in all respects? And then it gave me these trees here. So for instance, if you look at the green bar, so the green bar is 1 tree, and as we see, it's kind of in the middle there of all of them in the 3 different distance measures, because our model can use more than one distance. By the way, this is just a reality check to see if the multivariate exponential was working or not. And then we ask, well, what's the maximum likelihood? What's the, try to optimize, try to find the centroid tree, the one that represents all the others, because all the trees, they are a bit different, right? Otherwise, we would have only 1 point there. So if you look at 2,000 genes of fungi, you have 2,000 different trees. But one of them is going to be closer to all of them, and that's the one that I marked there as in red. And I think that's it. Yeah. So the reviewers always ask us to show that it's better, and I don't think it's a good way of thinking, because you can show there's a different way of thinking about the problem, even if right now it's not the best. And at that point we found, our method is best, but it's very close to another one that it's much faster than ours. It's a parsimony method. And so, in summary, so this is what I usually do. I find biologically motivated distance between the trees or between graphs, between anything. And then we use this distance to constrain the set of solutions, to regularize the solutions. And I think that's it. Yeah, thank you.
Hey, thanks for that. So if anybody online has got any questions, either pop them into the Q&A or raise your hand, and I can see if Teams will let me unmute yourself. Do you have any questions from anybody in the room first? If not, I have like about 6 I can pass around and you can ask them individually. All right, okay, I'll start off then. So could this be useful for things like drug discovery? So you're talking about different types of fungus, now we're all that connected. So things like the next penicillin or anything like that. Not just drug discovery, but are you using these trees to find things which are similar to things we like in different areas?
Yeah. So I wasn't thinking about drug discovery, but for instance, if you remember that all the antimicrobials, most of the antimicrobials, they come from bacteria, and so there's a gene for them. And so these methods, they can be used to reduce the uncertainty, because perhaps the gene is very short, perhaps the gene is small, the feature matrix is very small. And so if you have a very small alignment, if you have a very small gene, anything goes, right? It can say, well, perhaps it's this tree, perhaps it's this other tree. As I said, the uncertainty on the tree is very large. And we can use this method, even adding genes that you're not actually interested. I'm interested in this gene, but if I analyze all of them together, perhaps we're going to decrease the uncertainty on this gene that I'm interested, which are the antimicrobial resistance genes, virulence genes, or the peptides producing genes. So yes, I think anything that we know which genes are relevant, we can use there. And so the first thing would be to reduce the uncertainty. And there's another thing, which is, since these distances, they have a biological explanation. And then I think out of the bat, we can look at genes that have a lot of horizontal gene transfer, because those are the ones that horizontal transfer, is direct transfer from the gene from one bacteria to another one. It's not horizontal. It's not because there's it's not because it's my mom. It's something unrelated, and then I transfer to them. It's like how languages evolve. Languages, they evolve more through horizontally transfer than through vertical inheritance.
What's the actual mechanism of that? Like, are they like?
In the case of bacteria, it's plasmids. So plasmid is like a small chromosome. It's like a small genome that is there, and it acts. I don't know if biologists would like me to say that, but they act like parasites or viruses, in the sense that they have their own thing going on. But actually, there's a mutualism. They use something from the host, and the host usually use stuff from the plasmids as well. But yeah, it's a small thing, circular thing that is in the bacteria, and then it moves to another to other bacteria.
Mm-hmm. Just like leaning that into the uncertainty question, I was thinking about the evolution like early humanity, like sort of spreading out going to the rest of the world. Like you always see like news stories saying like, oh no, they turned left at this point, or no, they turned right at this point. Have you looked into any of that kind of work? And what I think would be really nice is to actually see like uncertainties on these kind of things. Like, based on this evidence that we have now, there's a 50% chance that this is what happened, or even looking at species tree in general, right? It's saying like this is what we think the tree of life is to this percentage accuracy. Like those things never seem to be communicated. data?
Yeah.
Is that something you can do?
Yes. Not with what I presented here. So usually I avoid working with human data, human evolution, because there's a lot of variability and it happened very quickly. So usually it's not the same time scale. I think it's more of an ecological scale than what I usually think. I think more in terms of evolutionary.
So generations?
Yes, exactly. Yeah. So you have, so the population process is very important.
Yeah.
So you have to think a lot about the population process. And I think, but not very deeply, I think just a little bit about usually you need convoluted models to describe. One thing that I used to say a lot is that 1/3 of our genes. So we know that our tree is like we, chimps, and then gorillas is an outgrowth. So we are closer to chimps than to gorillas. But 1/3 of our genes are closer to gorilla than to chimps. And that's because evolution happened so fast and the ancestral populations were so large that, so you have to remember that there was a population of individuals there, and some of them became what nowadays are the gorillas, and some of them became what nowadays are the chimps and humans. But at that point in space and time, they had different copies of the gene, and so their genes, the ones that end up becoming gorilla still have a copy that looks like the one that we have. So that's a case where some people don't like the term ancestral polymorphism, but you see a lot in the book. So they say about this. Ancestral polymorphism means that the ancestral species had polymorphism, which means more than one copy of the thing. And in human populations, you had a lot of this. So that's a hard problem for me to think about. And most certainly you cannot use trees. I remember that I think that the rate of recombination was the same as the rate of evolution. When you look at short time scale in humans, because every time, our mom and pop genomes, they get mixed and then it becomes, so that's the same effect as homologous recombination.
You're talking about simulations of, you can sort of simulate what genes would look like given a tree, and then you try to see if you can get the tree back out using the method. Could you go a step beyond that? Like, could you have like a simulation which is simulating the mixing of genes and stuff like that? So you have a step above.
Yeah.
You don't impose a tree, that you basically kind of game of life.
So I don't have here. So this was simulated, the lower part here was simulated using a method where we say that the loci is the plural of locus, so you have the locus, which is one region in the genome, and then the loci evolve, and they evolve through duplications and losses, and then once they evolve by duplication and losses, which is like a birth-death process, and then what happens there, it's a coalescent process. So the coalescent process is a very involved statistical technique. And my colleague was the one that actually calculated, because at that point, I think we knew for one species, the coalescent process for one species. But then when they become different species, what's a different species? Is when you stop exchanging material freely with them. And so that's the biological definition. I mean, the evolutionary biological definition of species are things that can freely exchange genomic between them. And when you stop this, it's like a pipe. So if you have two different pipes, the water that comes here, you don't know if the water is in one corner or the other of the pipe. The water is, the water molecules are free. But if you have different pipes, and then you cannot exchange them anymore. And so what happens inside the pipe, it's the coalescent process. And so you have models that describe this. So for instance, if the pipe becomes larger, and then you have a larger population, and then so you have more things that can change. So this was incorporated into the simulation model. but not in the estimation model. So the simulation model was very complete, and it's more because we want to see, it's just a reality check, right? If you simulate exactly as in your model. But in our case, we had a model that was much more complex, and then we wanted to see that. So yeah, simulating, we can do the simulation. For humans, I think the challenge would be to set the parameters, because we would need to see, from literature, what are the parameters for the probably no duplication and losses, and then the population sizes, how do they change over time? This will be challenging, but yeah, it could be done.
We might be quite envious about some people in the room then. So these are all, well, mostly astronomers. They have simulations where you can just say, make a universe. Right? Then it's going to go, and then you have different stellar populations kind of being born, galaxies kind of colliding, bigger galaxies forming in situ, ejector, all that kind of stuff, and you can compare that against what's going on in real life. It'd be interesting if you could make I don't know, just like a species simulation where you just like let the population just like go mix genes.
Yeah, I think you're talking about what people call forward simulation.
Like Sims, I don't know, just like the Sims but with genes.
Yeah. So forward simulation is to simulate things that we don't have yet, right? Simulate how it would look like in the future. We can do this in the short term and under some models, and then we can look if in the reality something happened. So for instance, I think for SARS-CoV-2, you can simulate how SARS-CoV-2 is going to look like in a few weeks from now. And then if you find something that is out of this distribution, and then you know that something happened there, some selection or something. But longer term, it's hard to simulate because we don't know they say that evolution is not teleological. Teleology means that there's an end, there's a finality on what we have. And so biological evolution, we don't know what's being selected, and it's being selected at the time that the organisms are reproducing. So we don't know if it's going to select for something or something else, and that's when the simulations fail. But you can simulate under a completely neutral scenario. Nothing happens, everything is normal, and then you simulate, and then you see if nothing happens, this is how the sequences are going to look like.
You can see kind of like simulate an asteroid coming in, wiping out all the evolutionary niches, things explode again.
This is kind of the founder effect. So the founder effect is when you have a population, and then this population decreases to the individual level, and so that's the founder. And then whatever this founder has, all the population will have. So
okay. So that's why you get like, Genghis Khan is the ancestor of like 10% of Europeans or something.
Or if you look into some tribes, everybody in the tribe has the same, I don't know, eye color or some,
Cool, really interesting stuff. All right, assuming there's no more questions, thank speaker. Thank you. So we'll be back next month with Sofia Valcosa, who is a researcher at CERN, talking about the Quantum Technology Initiative at CERN. So we'll see you all then. Thank you.